Hi, I'm Marianne, and I'll be discussing a recently published paper titled Detectability of Varied Hybridization Scenarios using Genome-Scale Hybrid Detection Methods. This work was completed at the Wisconsin Institute for Discovery at the University of Wisconsin. Phylogenetic networks extend phylogenetic trees to display information about possible hybridization, admixture caused by forces like horizontal gene transfer and integration. In order to find these hybrid relationships, there are many methods available to evolutionary biologists. Some methods reconstruct entire networks, and others find individual hybridizations between subsets of taxa. In order to aid those interested in applying phylogenetic methods, we reviewed and tested individual hybrid detection methods, Ticker, MSC quartets, Heidi, Patterson's D-statistic, DP, and D3. Each method has its own unique approach, using gene trees or sequence data in order to identify hybrids. For example, while Ticker and MSC quartets rely on quartets of taxa and gene tree topologies, methods like D3, Heidi, Patterson's D-statistic focus on site pattern frequencies or pairwise distances. So we simulated hybridization scenarios that varied in depth, quantity, and rate of gene flow. The variability in hybridization scenarios was chosen to identify hybridizations that might elude detection, as well as to compare method performance on hybridizations that may be more difficult to identify. These methods were compared in terms of precision, false positive, and recall rates. Some notable findings included that the deeper or more frequent hybridizations often generated noise, leading to a higher rate of false negatives across all methods assessed or lower recall, meaning that these methods are less likely to correctly identify hybridizations when multiple events occur or when they are further in the past. High precision in method performance was particularly noted with MSC quartets, which was consistent in identifying hybrids relationships under most simulated scenarios. Heidi also emerged as the only method capable of identifying both hybrid and parental lineages, which is invaluable for evolutionary studies. We also tested these methods on an empirical data set of ultra-conserved elements from the Nominae B subfamily to demonstrate its performance on real-world data. We found that regions of poor support in a tree previously attributed to gene tree estimation error could also be explained by the presence of hybridization. This paper provides a framework for phylogeneticists to select the appropriate method based on the available data of their study organisms. And I encourage you to review the paper in your own application of these popular methods to your genetic data sets and interpretation of their results. Also available is a GitHub repository, which contains and describes the simulation and data analysis scripts. Thank you for listening.